Hello, I am Rob David from Thermo Fisher Scientific, and this is Double Helix Diaries. In this episode, meet Dr. Steven Jackson, or Steve, as he is fondly known. He is an application scientist, senior manager at Thermo Fisher Scientific. Today, we're going to discuss the synergistic potential of combining two powerful technologies, next-generation sequencing and capillary electrophoresis, which he often describes as 1 + 1 = 3. Steve, it is so great to have you here with us at Double Helix Diaries. And in particular, I've been very excited to have this discussion as my roots in molecular biology were with Sanger sequencing. It was the very first technology that I worked with when I was in the lab as a technician.
Oh, well, thank you very much for the invitation. I'm happy to be here.
Your background is impressive. What we'd really like to hear is a bit more about your journey, how you got here. Could you share a bit more with me?
Sure. And by the way, thanks for the impressiveness part of it. But anyway, I've always been interested in genetics. I've always liked genetics. I had a research program at the University of Washington working on fruit flies, so genetics has always been very close to my heart. And as the genome projects, got underway, first it was yeast, then the flies, then the worms, then humans, all got sequenced together. Tremendous amount of genomic information became available. And I've always been interested in trying to understand how that produces, us, or a fly, or a worm, or a yeast, for example. And so finding out the links between the genome and the phenotype is always an interest to me. And of course, to do that, you have to understand the sequence. Next-gen sequencing has been, at the forefront of some of that. But of course, next-gen sequencing is fantastic for discovery-based, but you really want to focus on certain things. You really have to kind of delve into particular regions. You really want to understand variation in a very specific region. Sanger sequencing is very important for that. And of course, there are other tools that are important for other aspects of it, but Sanger is really a great tool for really focusing in on particular regions of a gene and variants that arise in that region.
So on to our next question. Scientists frequently engage in debates regarding the relative strengths of Sanger and NGS. If you could you elaborate a bit more on those relative strengths?
Sure. So next generation sequencing is an incredible technology. It really has modified the way that we understand biology. And it really excels at discovering sequences, really looking for new things, looking for entire genomes. You want to scan a very large set of genes or a very large transcriptome, for example. Next generation sequencing excels at those discovery-based explorations. You want to just look at new sequences. You want to look at an entire genome. You want to look at everything. Next generation is great for that. The downside to that is that you generate a whole lot of data that has to be managed. You have to be able to interpret it. You have to store it. You have to be able to extract it. So with that incredible amount of data comes an incredible amount of effort needed to really exploit that data. Sanger sequencing is sort of a complement to that because it allows you to focus on a region or regions. If you do multiple reactions, with a very high accuracy and specificity. So next generation is fantastic for doing discovery-based experimentation. Sanger sequencing is very good for focusing on regions or genes that you want to understand in more detail and with higher accuracy.
That reminds me, one of the, often when I was working with customers and fellow scientists, to me, the biggest benefit also was you mentioned, that discovery aspect of NGS, but then there's also afterwards, then we're wanting to confirm our discoveries and then focus down.
Yeah, that's an important thing because although it's gotten a lot better, there still are times I've seen lots of customers, and I go to meetings that are looking at some of these inherited diseases where they're doing a whole exome sequencing experiment to find out, for some developmental abnormality. They do a whole exome sequencing, they find some variants that are there, and they confirm those variants are real by using Sanger sequencing. So Sanger sequencing is a part of this discovery because they confirm, what has been found from the the huge fishing net, where they're like taking everything in.
Exactly.
Sanger sequencing is more like taking a small net and throwing it into the sea, and catching just a few fish, but very cost effectively and with very high accuracy.
I like that. I like that analogy. It's making me a little hungry too. I think I'm going to go out for a lobster dinner or something. We have a couple of follow-ups on this very topic. So we talked about these relative strengths, and we talked a little bit about some complementary aspects. Confirming. Can you elaborate a bit more on other ways that capillary electrophoresis and NGS complement one another?
Yeah. So one of the things that Sanger sequencing does is provides a very high accurate sequence read. Okay, so I like to say the peak is the peak. You see an A, a C, or a G, or a T. It doesn't depend on the algorithm used for lining up to a reference genome. Doesn't require a reference genome at all to line up against. So Sanger sequencing allows you to really kind of say, yes, that A is there, or that mixed base showing a heterozygote or tumor heterogeneity. That mixed base is indeed real. So that's where Sanger is very good for that. I think another aspect where Sanger sequencing really can play an important role is looking at methylated DNA sequences. So next generation sequencing again can do this. You can take a bisulfite converted piece of genome and do a next gen sequencing on that, but you have to build a reference database for the bisulfite converted sequence. You've changed the sequence completely, so you have to have a completely separate reference database, to compare against. And that reference database, if there's differences in methylation patterns, you can have different CG islands that are differentially methylated. You have to build separate databases for each one of those. Sanger sequencing simplifies that a lot because you take your bisulfite converted DNA, subject it to a regular Sanger sequencing reaction, and again, the peak is the peak. You can tell whether or not that A or that C has been changed to a T, and that you have a methylation state change there, without having to go through all this rigmarole of having different databases, different reference sequences that you have to build for that. So I think that's a growth area for Sanger sequencing, is that methylated DNA analysis, identifying biomarkers that are methylated sequences, looking at possible promoter sequence changes that affect expression level changes. Sanger sequencing, can play a very important role in that.
No, and I'm so happy you touched upon that topic. One of the questions I was going to ask about was metagenomics, and in particular methylation, and because it almost seems that, and this harkens back to another point that you were making. There's this big net that we're casting, and I think we were all very enamored of what next gen sequencing could do in that respect. But I feel the pendulum has swung a bit more back to realizing that very strength that you were mentioning for methylation in particular.
Well, and they're different tools to solve different problems. Next gen sequencing, long read sequencing, they're incredible for looking for new sequences. But to really confirm, to really to understand a particular region, Sanger sequencing might be more appropriate. In other words, you can think of next gen sequencing as a jackhammer, where it just like can tear up the entire genome and analyze that. But you wouldn't use a jackhammer to hammer in a thumbtack. Okay, so Sanger sequencing is more like the thumbtack, where you just want to have the most efficient tool for looking for accurate, cost-effective sequence determinations. You don't need a jackhammer all the time. Sometimes a thumbtack is entirely appropriate. And one more thing I want to say is that I mentioned these long-read sequences, and long-read sequencing technology again is a very important tool for looking at phasing between different alleles, for looking for chromosomal structure, that sort of thing. Again, it's very important. But even though, again, they're getting better at accuracy, when it comes to uncovering new variants, you still would want to use Sanger sequencing to confirm those new results that come off of a long-read sequence, because there can be inaccuracies with the way that the long-read sequences take place. So Sanger sequencing again is the preferred avenue for confirming that the variant is present, because again, the peak is the peak. That's all there is to it.
Indeed, well said. So, a bit of a different approach here with and see if we can discover a bit more together. What areas does Sanger sequencing excel, and where next generation sequencing might have limitations?
Again, it kind of comes to the simplicity and accuracy of Sanger sequencing. So, um, because Sanger sequencing can read through and produce a trace of nucleotides, Sanger sequencing can sometimes read through difficult regions better than next generation sequencing can. For example, homopolymers. Sometimes next gen technologies have a bit of a problem reading through G-rich sequences or homopolymers of A's or something like that. So Sanger sequencing does a little bit better sometimes, especially with some of the proprietary reagents that we can add to a reaction to get uh, um, read through of those difficult homopolymeric regions a little bit easier. Another important avenue of this is like adeno-associated virus that's commonly used in gene therapy research. And when customers are making their AAV-associated constructs, they're inverted repeats in the AAV genome, which are sometimes difficult to sequence through. And Sanger sequencing allows them to read through that a lot more easily than with next generation sequencing, because some of the hairpins, the things that next gen has a difficulty going through, are more easily read through by Sanger sequencing.
Correct me if I'm wrong, though. We also, as Sanger has been, is now a well established technology. We've done a lot of engineering of the polymerases that we use for sequencing as well. And my understanding is that that also lends to the accuracy of the
It
technology.
does, yes, and we continue to do that. So we continue to revolutionize the sequencer, the sequencing enzyme.
So to that end, I know that we now have we've done quite a bit of development on the instrumentation itself, having launched a number of newer sequencers in recent years. Can you, tell us a bit about them?
Um, uh, so this is going to really sound disingenuous, but I love the Seek Studio Flex. It's so much easier to use than some of the other platforms, and provides a lot of advantages that the older platforms don't have. So, especially if you're going through a big plate where you're trying to confirm a bunch of new variants that you want to confirm that you identified by a discovery-based approach. You need to confirm them. The Seek Studio Flex, allows a continuous throughput, a very easy user interface. It's just great for really, for I wouldn't say very high throughput, operations, but for mid throughput, continuously cycling 96 well plates through it, it's a great instrument.
So one of the things that I am recalling when, back eons ago when I was again on the bench, is that one of the differentiators for Sanger is long read sequencing. Can you tell us a bit about those aspects of the technology?
So I think long reads now is meaning something different than it did eons ago when both you and I were in the laboratory. So um, some of the next gen platforms, do 200 to 400 base pair reads. Okay, and so that's we can call that short read. Sanger sequencing can do that, but it can also go up to about 1,000 to 1,200 base pairs. And again, that's a great option if you want to focus on that region, look for phasing, look for multiple variants within that region, without having to break it up into 200 base pair segments and analyze it that way. So Sanger sequencing, the enzymes that you talked about, the chemistry, the instrument itself can read up to about 1,100 nucleotides, 1,200 nucleotides in a single reaction. So again, that kind of allows you to do certain things that some of the short read next gen can't do. Long read now, though, also refers to some of the other technologies that are single pore, single molecule reads, and in those cases they can get up to 10,000 to longer reads in a single reaction. So Sanger can't do that, and we've tried some other things, that to help with that, and right now, that's an avenue that we're looking for. We don't know if we're ever going to get to that length of a read, but for that again focus range between, say 100 and 1,100 nucleotides, Sanger sequencing excels at that.
And to that, building upon what you just mentioned, one also retains a very high accuracy from those first few reads, excuse me, the few bases through to that 1,000, 1,100,
Yes.
which is a differentiator as well.
That's right. That's right. That's right. Again, that they're getting better. The next gen technologies, they have in the past have had some issues with accuracy. They're getting a lot better, but Sanger sequencing is just intuitive and accurate. You look at the trace, you can tell what's there, and you can tell what is real.
And I would say also it's very satisfying too, seeing those peaks right in front of one.
Yeah.
Yes. So we have discussed a range of topics. One thing we haven't touched upon, though, is what excites you most about what is coming in the future for Sanger.
Yeah, so I think one of the things that people don't really get is that Sanger sequencing can be used for a lot of different things. One of the things that I'm working on right now is liquid biopsy applications with Sanger. And for this, we're looking at methylated DNA changes detectable in liquid biopsy samples from breast cancer patients. And to do this, I have a panel of some genes that I'm starting to kind of query, to see whether the methylation state changes in liquid biopsies from breast cancer. And from that, we can maybe see, if, for example, there are changes in the promoter of BRCA1 that is causing it to be downregulated, leading to a particular type of breast cancer. Can that be detected with Sanger sequencing and liquid biopsy? I'm working on that right now. I know that it is possible, and right now I'm just trying to optimize the conditions and the protocol. So I think that this is not just for breast cancer, but looking at methylated markers in liquid biopsy samples from colon cancer, from any other types of cancers, or even from other kinds of diseases. I think that's very exciting because liquid biopsy is revolutionizing the way that we analyze pathologies, and I think if Sanger sequencing is shown to be a big part of that, it'll ease our customers' workflows and data access a lot.
That's really exciting and I think gratifying to hear too, because at the end of the day, why are we all here? We're here to make the world healthier, safer, cleaner, and that's really speaks to it.
Yep. Great.
Well, Steve, I think that wraps it up here. I just want to say thank you very much for being here on Double Helix.
Well, thank you. Thank you for giving me the time to talk about this. It's obviously it's something that's very dear to my heart, and so I'm happy to evangelize. So thanks again for giving me the opportunity.